This video is going to be all about preparing cells to be viewed under the microscope, and then two different types of staining that we can do to visualize them. There's actually a lot of other specialized types of stains that we're going to talk about to look for specific structures on an organism, like a capsule or an endospore, flagella, things like that. There are more specialized ones, and we'll only talk about a couple of them. But for simple stains and negative stains, they're really just to get a quick snapshot of the organism, okay? So most microbes are colorless or translucent. So if you were just to go and collect a random water sample and you put the water on the microscope slide and stuck it under the microscope, you wouldn't really be able to see a whole lot, right? Because everything is colorless. And so we need to provide contrast in order to visualize these colorless or translucent microbes. And we do that by adding a stain. But before you can go staining a sample, you need to make sure the cells you're wanting to look at are actually stuck to the microscope. And so that's going to be called making a smear. And with the types of stains that we're going to talk about today, again, you just get a snapshot of the organism. So you'll be able to tell the morphology of the cells and the arrangement of the cells. Now, with a negative stain, you'll also be able to tell the cell size, assuming your microscope has like a little ruler embedded in it. There are some microscopes that one of the lens just always has a small ruler inside, so you can actually determine the very specific length of an organism. So in order to make a smear, you're going to need your microscope slide, some water, and your culture, and then the stains. Now, I'm not going to quiz you on what's required. I'm just these are just some of the more important items. You'll also need your Bunsen burner to do aseptic technique. Some labs use air dryers to help speed up some of the steps. But the thing you really need are the cells you're going to stain, your inoculating loop, and then the stains. Those are the real important things. You can't make a smear and do staining without those materials. So when it comes to stains, when we're talking about the simple stains are basic dyes, okay? That means they have a positive ion associated with the color, okay? And so all bacterial cells have an overall negative charge. That's just part of their cell wall structure. All bacteria have a negative charge associated with them. So when you add a positively charged dye or stain, that colored stain is going to be attracted to the entire surface of the bacterial cell, which is how you're going to be able to easily see its morphology and arrangement. Okay, so three common simple stains that are used in the lab are crystal violet, which stain the cells violet, as you see on that bottom left picture. Safranin turns cells red, and then methylene blue, as the name implies, will stain the cells a blue color, as you can see on the far right. So remember, these three stains are simple stains, which means they are basic stains, so they have a positive charge associated with them, which is attracted to the negatively charged bacterial cell. So if you look at these pictures, notice that you get this white colorless background and very colorful cells, okay? That is the hallmark of simple staining, a whitish background and stained colorful cells. Okay, now depending on how well the staining was done, kind of like in that middle picture, there's sometimes kind of a color associated with the back, but if you wash your cells and do your staining completely properly, you shouldn't have any residual color in the background. It should always be the white background and colorful cells. Okay, so let's talk about how we actually go about preparing a sample and then staining it. So first you want to you create the smear, okay, which is where you're going to put the bacterial cells on the slide and attach them to that slide, okay? So the exact procedure varies depending on which type of media your starting culture comes from. If you're using a broth, right, it's already a liquid. So you can literally just flame your loop and using proper aseptic technique, transfer the bacteria from the test tube onto the microscope slide. Now you're going to want to kind of spread those cells out on the microscope slide because you want to make sure you can view individual cells. If you were to just put a bunch of cells in the same spot, they're going to overlap and then be all crunched up against each other, and it's going to be really hard to determine their arrangement. So you want to kind of spread your cells out on that slide, and then you need to let it air dry. And this is the point where it's okay to use little air dryers, which just kind of speeds up the air drying process. After that, you're going to heat fix the cells. And what that means is you have your Bunsen burner on, and we usually use a clothespin to hold one edge of the glass microscope slide, and then you pass the rest of the slide through the Bunsen burner 3 times. And you can see this procedure on the other PowerPoint about making smears and stains. Okay, there's a video of this process. And then once you've heat fixed your slide, you're ready for the staining process. So the smear process stops after heat fixing. Now, you might be asking, why are we passing this slide through the flame 3 times? And that is so we can A, kill the cells, B, make them stick to the slide, and then 3, make them take up the stain a little more easily. Okay, so again, that was the process if you're starting from a broth culture. Now, if you're not starting from a broth culture and you're going to be taking cells from an agar plate or a slant, then the first thing you're actually going to want to do is put some sterile water onto your slide, just a tiny drop or a loop full. Some people like to squirt water onto their inoculating loop and get just a loop full of water. Put that on their slide, and then you will aseptically transfer cells from either the slant or the agar plate into that water droplet. And then you're going to, again, smear it out. And then you're going to let the slide air dry and then heat fix it. Okay, so it is really important that you are patient on the air drying step, because if there's any water remaining on the slide when you go to heat fix, then passing that little bit of water through the Bunsen burner is essentially going to boil your cells. Okay, and that can just completely disrupt the shape and morphology in your entire sample. Okay, so you don't want to boil your cells. So you have to be really patient and let them air dry completely. Okay, all right, so once you've created your smear, you are going to place the slides over this little staining rack that you can either have like over a pan or over the sink, basically so you can just pour a bunch of stain, whether it be methylene blue or safranin or crystal violet, and you just completely soak the slide, and then you wait, and it's normally 30 seconds to 1 minute is sufficient. And then you still have that clothespin on your slide, right? So you don't get stain all over yourself, and you kind of give it a little flick to try and get any excess dye off. And then you are going to use a spray bottle and very gently, starting at the top of the slide, rinse it. You don't want to blast your cells with a big stream of water. Okay, it would be kind of like blasting us with a fire hose, right? So you want to squirt the water so it hits the top of the slide and just gently runs down. So it's rinsing the slide without disrupting the cells that you've attached to them. And then you can either kind of like give the slide another kind of little flick, kind of get the excess water off. You can dry it with bibulous paper if you want, but then it's ready to go under the microscope. Okay, now you're usually going to end up using the oil lens, so you're going to want to have your immersion oil ready for that process. All right, so that's what you do for a simple stain, okay? So again, you'll have that nice white, crisp background with colorful cells, and you'll be able to easily see the morphology and arrangement. Now, you cannot determine the size of these cells because the heat fixing process causes cells to kind of go and kind of shrivel a little bit. So the exact size of them is altered when you pass your slide through the Bunsen burner. And so if you need to know the exact size of your cells, you're going to want to use the negative staining procedure. And so the negative staining procedure is relatively simple as well, but this process does not use heat fixing. There is zero heat fixing in this procedure. So we need to be able to visualize the cells as they are. And so how we do that is we use a negatively charged dye. Okay. And so what happens is by using a negatively charged dye or an acidic stain, you are going to introduce this negatively charged colorful stain to your negatively charged cells. Well, what happens when the charges are similar, right? They're going to repel. So the negative stain is opposite of the simple stain in how it works, but also in what you see. As you can see in this picture, you're actually going to get a more colorful background, and these cells themselves are going to appear colorless because the dye is repelling the cell. So the background gets stained, but the cells remain colorless, allowing you to still see the morphology and the arrangement. But again, the cell size has not been altered because they haven't been exposed to high heat. Okay, so you use this approach when you need to know the specific cell size, but also if you're dealing with really delicate cells like spirochetes. Spirochetes are super thin. And so if you were to try to do a simple stain, when you expose those spirochetes to the heat, it would like obliterate them, okay? It wouldn't be any good. You wouldn't get any useful information from them, okay? So the procedure is a little bit different because there's no heat fixing, but I'm not going to ask you to memorize the steps of this procedure. I really just want you to know why you would use negative stains and kind of what you would see under the microscope. And then once you're done with smears and staining, okay, anything that had living organism on it, like your slides, needs to go into the bleach solution. You're going to always wipe down your bench with either the Lysol solution or ethanol, depending on what's available. Hang your aprons in the back and then throw your gloves in the trash can. I did post an optional lab report. It's just good practice to go through and see what types of questions you might see on the exam and help you kind of assess whether or not you really understand the theory behind staining and identifying a simple stain versus a negative stain. So the lab reports are not for a grade. It's merely for additional practice applying what we're talking about in these videos. So if you have any questions about the content or anything in the lab report, please send me an email or a text. I'm happy to walk through it with you. Bye, y'all.